So, uh, if you are anything like me, you have probably found yourself staring at your phone late at night, just endlessly scrolling through health news.
Oh, absolutely. The algorithms definitely know what we want to click on.
Right. And you pause on those wild, almost unbelievable headlines. You know the ones I'm talking about? The articles promising the absolute holy grail of biology, a magical way to completely reverse brain aging.
Yeah, the fountain of youth clickbait.
Exactly. Because, I mean, we all want to keep our minds sharp. Nobody wants to lose their memories or their cognitive edge as the decades pile up. And the media ecosystem knows exactly how to tap in to that deep, universal desire.
It's a very primal fear, losing your memory.
It is. But today, we are going to bypass all of that internet hype. We are going to look directly at the actual science behind one of the most talked about biological discoveries in recent memory. We've got a fascinating deep dive ahead of us today, focusing on a landmark study. This was published in the journal Nature Aging back in August of 2025.
Yes, and this is research coming out of Saul Vallada's laboratory at the University of California, San Francisco. It is genuinely turning heads in the neuroscience community right now.
And for good reason. They essentially found that reducing a single iron storage protein in the brains of aged mice didn't just, slow down cognitive decline, it actually reversed it, bringing their memories back to useful levels.
And we really need to emphasize that the word reversal is incredibly heavy in the field of neurobiology.
It's not a word thrown around lightly. Right.
Not at all. Usually the best we can hope for in aging research is to hit the brakes, just slow the decline. To actually put the car in reverse and drive back to a state of youthful function is the ultimate goal.
Yeah.
But while the findings out of UCSF offer genuine hope and provide a remarkably clear mechanical story about how our brains age, there is a massive gap between early data in a laboratory and the master switch narrative that the media immediately ran with.
So true. And our goal in this deep dive is to explore what the science actually demonstrates, step by step, and where this discovery truly stands today, free of all that commercial noise.
Exactly.
We definitely want to equip you with the facts so you can spot the difference between a real scientific milestone and a snake oil sales pitch. So let's start at the very beginning of the UCSF team's journey, which was led by first author Laura Ramizel.
Right.
If you were a scientist trying to cure brain aging, how do you even know where to look? The brain has, thousands of different proteins and genes operating at any given second? Did they just have a hunch about this one specific iron storage protein, or did they have to go fishing for it?
They cast an incredibly wide net. In science, we call this an unbiased approach. They didn't start with a specific assumption.
They just looked at everything.
Pretty much, they took the hippocampal tissue, that is the region of the brain responsible for learning and memory from young mice. These are basically mouse teenagers, about 3 months old.
Okay.
Then they took the same tissue from aged mice, which are around 18 to 24 months old, and they used highly advanced sequencing tools to look at all the genetic instructions being read by the cells, as well as the actual physical proteins being built.
The transcriptomic and proteomic data.
Exactly. They essentially took a massive, comprehensive snapshot of a young brain and laid it right on top of a snapshot of an old brain to see what was different.
And out of that massive comparison, looking at all those overlapping genetic markers and proteins, they were looking for the needle in the haystack. And a singular culprit emerged.
It really did stand out.
It's called FTL1, which stands for ferritin light chain 1. And the detail that really caught my attention here is that FTL1 was the only protein that consistently increased with age across every single data set they looked at.
The only one.
Yeah, the genetic instructions for it were elevated, and the physical protein itself was sharply higher in the older brains. I mean, the paper noted a p-value of less than 0.0001. It wasn't a subtle shift. It was a massive, undeniable spike.
Yeah. And that level of consistency is rare when you are looking at something as complex as aging. Usually you see dozens of proteins slightly shifting up or down, creating a very noisy picture.
Right, a lot of static.
Exactly. So seeing FTL1 stand out so clearly told the researchers that it wasn't just a bystander, it was deeply entangled in the aging process of those memory-forming neurons. But as we constantly have to remind ourselves in science, just because a protein is present at the scene of the crime doesn't mean it committed the murder.
Correlation is not causation.
Precisely. Just because FTL1 is elevated in older brains doesn't automatically mean it is the thing causing the brain to act old.
Okay, let's unpack this, because this is where the UCSF team did the heavy lifting to prove their case. They used a bidirectional experimental design.
Which is the gold standard for this kind of work.
Right. That means they have to prove the concept in two opposite directions. First, they asked the crazy question, what happens if we take a young, perfectly healthy brain and force it to become old?
Making young brains old.
Yes. To do this, they took young mice and artificially forced their brains to produce way too much FTL1. How do you even do that without hurting the animal?
You use what is known as a viral vector. You can think of a viral vector as a microscopic delivery truck. Viruses are naturally very good at breaking into cells and delivering genetic payloads, so scientists hollow out a harmless virus, pack it with the genetic instructions to build FTL1, and send it directly into the neurons.
So the young neurons suddenly start manufacturing this protein like crazy. What actually happened to the mice?
The results were striking. By simply adding more of this one single protein, those young mice effectively developed aged brains.
Wow.
On a microscopic level, the neurons started looking old. They showed reduced dendritic branching, meaning the little arms that neurons use to reach out and talk to each other started to wither.
They were losing their connections.
Yes. They had fewer synapses, which are the actual connection points, and behaviorally they failed. The researchers put them through standard cognitive tests like navigating a Y-shaped maze or recognizing a new object placed in their environment.
The novel object recognition test.
Exactly. And these energetic young mice suddenly showed the measurable memory deficits and confusion you would normally only see in an elderly mouse.
They successfully broke the young brains just by turning up that one dial.
Yeah,
That is fascinating, but also a little terrifying.
it is.
The second half of that bidirectional design, though, is the hopeful part, making old brains young. To do that, they went in the exact opposite direction. They took the aged mice, the ones that were naturally experiencing memory loss, and reduced their FTL1 levels. And they didn't just try one method to lower the protein. They used 3 completely independent genetic tools. They used RNA interference to block the genetic message. They used CRISPR-Cas9 gene editing to alter the DNA. And they used a conditional knockout method to just delete the function entirely. Why go through all that trouble to do the same thing 3 different ways?
Because scientists are inherently paranoid about flukes. If you only use one tool to reduce the protein, a skeptic could argue that your specific tool accidentally caused the memory improvement through some unrelated side effect.
Ah, I see.
By using 3 radically different molecular tools to target FTL1 and getting the exact same result every time, they made their findings practically bulletproof.
That makes a lot of sense.
And the results were remarkable. When FTL1 was reduced, the aged mice regrew those synaptic connection points. The branches on their neurons became complex and healthy again. And most importantly, their memory performance on those maze tests significantly improved.
I want to share a direct quote from Saul Villeda, the lead researcher, because scientists usually hedge their bets and use very cautious language. But he was incredibly blunt about what his team saw in the lab. He said, "It is truly a reversal of impairments. It's much more than merely delaying or preventing symptoms."
That is a very bold
To
statement.
hear a neuroscientist use the word reversal without a dozen caveats attached is just wild.
The reason he has the confidence to say that is because they didn't just observe a behavioral change. They mapped out the exact biological machinery underneath it. They found the why.
Here's where it gets really interesting. Because the mechanism they uncovered reads exactly like a mechanical failure in a car.
It does.
Let's use an analogy to make sense of the cellular biology here. Imagine your brain cells are powered by tiny, intricate engines. FTL1 is a piece of a larger storage container inside the cell that holds on to iron. You absolutely need iron to survive. It's a critical mineral.
Essential for life.
Right. But as these brain cells age and the FTL1 levels skyrocket, it's like those storage containers become faulty. They start leaking.
Yeah.
And when that iron leaks out, it begins to literally rust the engine of the cell from the inside out.
That is a highly accurate way to visualize it. To translate that into the biology occurring inside the cell, elevated FTL1 alters the oxidation state of the iron. It causes a buildup of oxidized iron, specifically F3+.
The rust.
The rust. And this oxidized iron is highly toxic to the cell's power grid. The power plants of your cells are the mitochondria. When the mitochondria are exposed to this toxic iron buildup, they start to malfunction.
They break down.
Yes, they can no longer synthesize enough of a molecule called ATP. You can think of ATP as the fundamental energy currency of the cell. It pays for everything the cell needs to do.
So it's a budget crisis.
Exactly.
Yeah.
If the mitochondria can't produce ATP, the neuron essentially runs out of budget.
Yeah.
It can't afford to maintain its synapses, it can't afford to send signals, and ultimately you experience that as cognitive decline.
It's a literal chain reaction. Too much storage protein leads to toxic rust. Toxic rust breaks the power plant. The power plant stops making energy. And without energy, the brain cell basically powers down.
That's the exact metabolic cascade.
But the UCSF team went one step further to prove this chain reaction was real. They essentially ran a rescue mission. They took these exhausted old neurons that were filled with toxic iron, and they treated them with a natural coenzyme called NADH. From my understanding, NADH is a helper molecule that directly supports cellular energy production. It acts like an emergency backup generator.
That is the perfect way to describe it. NADH bypasses the damaged mitochondria and helps force the production of ATP anyway.
And when they gave this backup generator to the neurons in the petri dish, the neurons completely woke back up. They started growing their branches again, restoring that neurite outgrowth in vitro. Even more incredibly, when they gave it to the living mice that had been engineered to have failing memories, their cognitive deficits were reversed in vivo.
What's fascinating here is that the NADH experiment serves as the ultimate proof of concept for the whole study. It proves that the metabolic pathway, the energy deficit, that loss of ATP, is the primary mediator of the memory loss.
It's all about the energy.
Right. If you bypass the toxic iron and just give the cell the energy it needs, you fix the memory, even if the FTL1 protein is still elevated. It is a brilliant, elegant piece of scientific validation.
But there is a catch.
There is a massive catch, because unfortunately, publishing that specific detail about the NADH rescue inadvertently opened a massive door for the commercial sector. It opened the door for exploitation.
Oh, we are definitely going to talk about the supplement scammers. But before we get there, let's play devil's advocate for a second.
Yeah.
If I'm listening to this, I might be thinking, okay, this is a beautifully told story about a rusting engine in a mouse's brain. But mice are not tiny humans.
That is a very fair point.
We've cured cancer and Alzheimer's in mice 1,000 times over, and it almost never translates to us. Does this FTL1 discovery actually have any bearing on human biology?
That is the most critical question you can ask when reading any preclinical science. The good news here is that the UCSF study didn't happen in a vacuum. It was built upon a very sturdy foundation of decades of human evidence linking ferritin and iron to our own cognitive decline.
So there's precedent.
A lot of it.
Yeah.
For instance, back in 2015, there was a major study published in Nature Communications by a researcher named Scott Aiton. He tracked over 300 human subjects for 7 years.
Oh, wow.
He found that the levels of ferritin in a person's cerebrospinal fluid, the liquid that physically bathes our brain and spinal cord, actually predicted how quickly their memory would decline.
That's a direct link.
It is. And this was especially pronounced in people who carry the APOE epsilon 4 risk gene, which is a known major risk factor for Alzheimer's disease.
So doctors were already seeing the smoke in human patients long before these researchers found the actual fire in the mouse cells.
Yes, the clinical data was pointing directly at this mechanism. We even have studies showing that if you look at the brains of Alzheimer's patients after they pass away, the ones with higher iron levels deteriorated much faster. But perhaps the most undeniable piece of evidence connecting FTL1 to human brain health comes from a very specific, rare genetic disorder called neuroferritinopathy.
I saw this in the notes. This is a disease that is directly caused by a mutation in the FTL1 gene itself, right?
It is. It is incredibly rare, affecting only about 90 known patients globally. Because these individuals are born with a mutated FTL1 gene, their bodies cannot properly manage iron inside their brain cells.
So the mechanism is broken from birth.
Yes. And as they get older, they suffer from severe, progressive movement disorders, muscle spasms, and eventually profound cognitive decline. As tragic as the disease is, it provides us with irrefutable, direct human proof that disrupting FTL1 and iron balance destroys human neurons.
It connects the dots perfectly between the mouse model and the human reality. It's also really interesting to look at the researcher behind this new study, Saul Valletta, and how his career has evolved.
He has a very interesting track record.
Yeah, if you follow longevity or anti-aging research, you might actually recognize his name from some wild studies over the last 10 years. Back in 2011, he was one who showed that physically connecting the circulatory systems of young mice and old mice, basically sharing their blood, rejuvenated the older brains.
The parabiosis experiments.
Right. Sounded like something out of a vampire novel. Then in 2023, his lab isolated a specific component in the blood called platelet factor 4 that seemed to be driving that rejuvenation.
Yeah.
But this new study on FTL1 feels like a massive pivot for him.
It represents a major strategic shift for his lab, and really the entire field. For over a decade, a huge chunk of anti-aging research was focused on circulating blood factors.
Looking for stuff floating around in the plasma.
Exactly. The idea was to find something in the plasma of young people that you could inject into older people to sort of wash the brain and heal it from the outside in. Targeting FTL1 represents a move toward fixing a protein that resides entirely inside the aging brain cells themselves.
So we're moving from trying to wash the engine with fancy oil to actually getting inside the cylinder and fixing the rusting parts.
That's a great way to put it. It is an intrinsic neuronal target, which is fundamentally harder to reach, but potentially much more powerful.
Which brings us to how the world reacted when this inner engine fix was announced. Let's talk about the hype machine versus scientific reality, because the way this study was portrayed in the media is a textbook example of how science gets distorted.
It happens almost every time a major paper drops.
The original press release put out by the university was actually quite measured. The headline was simply, "This protein slows the aging brain, and we know how to counter it." That seems perfectly reasonable.
It was entirely appropriate for the data they presented.
But then the science aggregators got a hold of it. The headlines escalated quickly. One site ran with, "Blocking this one protein restores aging brains." Another popular site pushed it further to, "Master switch brain protein could reverse age-related memory loss."
And there's that phrase.
Right. And by the time the story reached the health and wellness blogs, you had headlines asking, "Can one molecule rewind your mind's clock?" They were using sensational language that the actual scientists never ever used.
This highlights a systemic problem in how scientific news is consumed. Several major distortions happen simultaneously. First, almost every one of those viral headlines completely erase the species gap. By omitting the word mice, they implicitly promise their readers a human breakthrough.
And then you have that phrase, "master switch." The media is obsessed with finding a silver bullet for aging. But if you read the actual UCSF paper, they're very careful to call FTL1A pro-aging factor. They openly acknowledge there are likely dozens of other factors at play.
It's never just one switch.
But the most egregious fallout from all of this hype was the immediate commercial cash grab. Because the researchers used NADH to rescue the mouse cells in a petri dish, the supplement companies pounced.
They wasted no time.
Within days, a company called Herb Green Health started pushing content, framing their over-the-counter NADH supplements as a proven way to reverse brain aging, just entirely unsupported by human clinical trials.
If we connect this to the bigger picture, it is vital to listen to what independent experts say when these studies drop, rather than the people trying to sell you a bottle of pills.
Always follow the money.
Right. Andrew Steele, who is a highly respected researcher in the anti-aging space, offered a very sobering take to BBC Science Focus. He noted that this is fascinating early research, but added that there are a few steps between this and new brain aging medicines for humans.
Saying there are a few steps between injecting a virus into a mouse's brain and buying a pill at the pharmacy is a very polite British way of saying, slow down, everyone.
Absolutely. The longevity research outlet lifespan.io also provided a fantastic analysis. They pointed out that while the relationship between the FTL1 protein and memory in the mice was statistically real, it was not an absolute law.
What do you mean by that?
Having more of the protein meant a mouse was likely to perform worse on the maze test, but it was probabilistic, not deterministic. It is not an absolute on or off switch where turning it down guarantees perfect memory.
We also have to talk about the fine print, the limitations of the study itself. These are the details that those viral aggregators conveniently ignore when they broadcast the news to their millions of followers. What are the big red flags or caveats here?
The limitations are crucial, as they always are in preclinical work. First, they used only male mice in this study.
That seems like a big oversight.
In modern neuroscience, ignoring sex-specific effects is a glaring omission because male and female brains metabolize energy differently and can age quite differently. Second, they only looked at the hippocampus.
Just the memory center.
Yes. The brain is vastly more complex than just its memory center. We don't know what reducing this protein does to the frontal lobe or the motor cortex.
I also noticed in the methodology that the initial broad net they cast, the transcriptomic comparison that identified FTL1 in the first place, relied on a tiny sample size of just 5 mice, 3 young ones and 2 old ones.
They obviously validated it with larger groups later, but yes, that initial discovery pool was very shallow. Furthermore, we have to look at how they delivered the treatment.
The viral delivery trucks.
Right. They used a stereotaxic injection to get those viral delivery trucks into the brain. That is a highly invasive surgical procedure where you drill through the skull to inject fluid directly into deep brain tissue.
Yeah, that is obviously not clinically viable for humans. You can't drill into millions of people's skulls just as a preventative aging measure.
Certainly not. And finally, there is the raw biological risk. Broadly reducing a protein that stores iron risks dangerously disrupting the brain's delicate mineral balance over the long term. If you overcorrect and remove too much iron storage, you could accidentally create entirely new neurodegenerative problems. Essential iron homeostasis has to be maintained.
So given all those caveats, the brain surgery, the male mice, the risk of iron toxicity, where does FTL1 actually fit into the broader race to cure brain aging? It feels like every single week I'm reading about a new miracle cure for dementia or cognitive decline. How does this compare to the other stuff in the pipeline?
It is an incredibly crowded and fast-moving landscape. To give you some context on the competition, you have senolytic therapies. These are drugs, specifically a combination of dasatinib and quercetin, designed to clear out zombie cells that stop dividing but refuse to die, which accumulate with age. Those are already in phase 2 clinical trials for Alzheimer's patients.
And what about the epigenetic reprogramming we hear so much about, the cellular rebooting?
Yes, the Yamanaka factors. That approach involves using gene therapy to partially reprogram older cells back to a youthful state. Companies like Youth Biotherapeutics are actively getting feedback from the FDA right now about starting human trials for brain gene therapies using that exact method.
So the competition is fierce.
It is. You also have metabolic drugs like GLP-1 receptor agonists, the same class of drugs as semaglutide, being heavily investigated to see if they protect neurons. And you have researchers looking at exercise factors like irisin, a hormone your body produces when you work out, to see if it can be put into a pill to mimic the brain benefits of hard exercise.
With all of those therapies already moving toward human trials, why should we care about FTL1? What gives this newly discovered protein an edge over the rest of the pack?
Its greatest edge is the sheer clarity of its mechanistic story. We aren't guessing how it works. We know the exact cascade, the iron accumulation, the mitochondrial disruption, the lack of ATP energy. And crucially, it converges perfectly with 2 decades of existing human data on brain iron and ferritin.
It isn't just a random protein found in a mouse.
Exactly. It's a target we already know causes human neurodegeneration. And the researchers are not resting on their laurels. Concrete next steps are already outlined. They are moving to test female mice to ensure it works across sexes. They are testing it in specific Alzheimer's disease models. And most importantly, they are looking for pharmacological FTL1 inhibitors.
Oh, drugs that can do what the viral injections did, but simply by swallowing a pill.
Right.
Like ferritin, perhaps. I know that's an iron-chelating drug that binds to excess iron, and it's actually already in phase 2 clinical trials for Alzheimer's and Parkinson's.
Exactly. Exploring pharmacological options like deferriprone means the pathway from this lab discovery to a tangible therapeutic lead is already being drawn out.
So what does this all mean? Let's synthesize this deep dive. The FTL1 study out of UCSF is an incredibly compelling single-target finding, using that rigorous bidirectional design to prove that one protein can both cause and reverse cognitive decline in mice is an amazing milestone.
It is. It proves the engine can be fixed.
But the distance between injecting viruses into the brains of male mice and you picking up an FDA-approved prescription at your local pharmacy spans years of rigorous work.
The ultimate cautionary tale from our discussion today is how rapidly commercial exploitation moves. The second those researchers published that NADH rescued cells in vitro, supplement marketers twisted that elegant science into a sales pitch. Critical thinking is our best defense against the hype machine.
So true.
FTL1 is a brilliantly identified, highly promising target, but it is not yet a proven human treatment.
I think that's the perfect place to land. We always like to leave you with a final thought to ponder on your own. The research we explored today suggests something profound. Cognitive decline isn't just inevitable wear and tear. It's not just your biological machine breaking down randomly because it's old.
It's an active process.
It's an active metabolic shift. It's a fundamental change in how your cells handle basic resources like iron and energy. So if aging is just a biological program that can run in reverse under the right conditions, what other essential daily functions in your cells are quietly changing their behavior right now, just waiting for science to find their master switch? Something to think about. Thanks for joining us on this deep dive. Keep asking the big questions.